Good morning. So, how are you all? Can you hear me? Okay, hopefully you have seen the midterm topics which are uploaded on the drive, and you know what are the quite uh, big material, but however, I think you have enough time and you can prepare very well for this midterm. So maybe you have questions regarding these topics. So today we will go through the tissue samples, of course, micrographs. Also, I will try to answer all your questions. Let's start. How many people we have today? Okay, 7 students. So you see that quite a lot of type of cells you have in this part of histology. So connective tissue, proper connective tissue, specialized embryonic connective tissue. So um, that is why hope you have to be able to identify these cells on the images, which will be included in your midterm. So I have to say that micrographs are included only in the midterms. They will be not used in the final exam. So let's start. Connective tissue proper. First of all, do you remember types of cells? What type of cells you have in this tissue? what type of tissues and what type of cells. Connective tissue itself has several subtypes: connective tissue proper, connective tissue special, and embryonic connective tissue. Okay, so what type of cell is the main principal cell? What type of cell is the main principal cell of the connective tissue? Cell which might be represented as an active or inactive, and plus it is able to generate and release, produce and release the, for example, collagen fibers. Okay, one idea is that they are mesenchymal cells. Okay, maybe another idea: what type of cells are principal cells which generates, yes, exactly, fibroblasts. Fibroblasts. What is the difference between the fibroblasts and fibrocytes? Maybe it is possible to, for example, Charlene, please, can you tell me what is the difference between the fibroblast and fibrocytes?
I
Fibroblast,
think, yeah, I
fibrocyte.
think the fibroblast
So
has
can you switch on
collagen
your
fiber.
phone microphone? I can't hear
Pardon?
your voice. Wait, wait, wait.
Okay.
Yes,
Shall I
please
write it down instead?
say it again, Charlene.
I think the fibroblast has collagen fiber,
What about the
and
fibrocyte?
the fibrocyte hasn't.
Okay,
It's like it's still in early stage of formation.
okay. So another idea maybe regarding the fibroblasts and fibrocytes, the main difference between the fibroblasts and fibrocyte. For example, from point of view of morphology. Do you remember how they looks like these both cells?
Fibroblast
Henriette,
has a very
please. Henriette, maybe you.
pardon.
Fibroblast and fibrocyte. Difference between the fibroblast and fibrocyte.
I think fibroblast has a big nucleus.
Big nucleus. From functional point of view, maybe Justin, you can you give me the answer?
Fibroblast is active, but fibrocyte is not active.
Yes, exactly. Uh, who is talking now? Oh, okay. How I have to pronounce your name? Sorry, Khader.
It's KHODR Khudur.
Yes, Khader. Okay. Am I right? Khader.
Yes.
Oh, so, thank you. So you
Yeah.
are absolutely right. So this is the main difference between these two type of cells. These are not different type of cells, or they are.
Sorry, repeat the question, doctor.
So fibroblast and fibrocytes, are they different types of cells?
No, they are the same, but
The
it's
same?
different.
Yes.
Yes, but different in activity according to the
Yes.
growth factor.
Exactly. So in this case, how do you think what type of cell organelles, for example, might be um, represented? in the fibroblast, which is not identifiable in the fibrocyte. I mean, main morphological characteristics of active cells and inactive cells. Who can characterize this? Should I repeat my
The
question?
the active cells have a large amount of rough endoplasmic reticulum, while the inactive ones have a few amount.
Yes, exactly, Precio. Yes.
Yes,
May, maybe what else? What else? What about the nucleus? Nucleus, how it looks like.
Nucleus, like there is like EU chromatin and there is like dark one.
So if it is stained dark, for example,
Yes.
nucleus. So I mean, when in the cell is prevalence of dark stained material, what does it mean? We have their prevalence of heterochromatin, euchromatin. Which one?
that's fibrocyte. The heterochromatin, the tightly bound chromatins, are found in the fibrocytes because they are not as active as the fibroblasts.
Yes. Okay. So why it is not so active as a, for example, euchromatin? Why
Because
heterochromatin?
the
Yes.
euchromatin is the stage. It makes cell division easier for the fibroblast.
Cell division. What about the gene expression?
Gene expression.
Yes, this is, everything depends on gene expression, even the mitotic division of the cells. So when the cell is active, the gene expression is active, and the cell is able to produce a lot of different types of proteins. So, okay, if I show you now the image of cell, can you you have to identify, you have to try to identify type of cells, okay? So I will go to the micrographs. What type of micrographs are these? Micrographs, look. What type of micrographs?
The electron micrograph.
Yes, exactly. So if they are electron micrographs, or 10 micrographs, yes, or EM micrographs, electron microscopic micrographs. So what you see here, what types of cells you can identify here. For example, number, okay, 3, it should be here somewhere, 3, but we miss it. Okay, this one, look, this cell. What type of cell is this? This is TEM,
Is
or
that a macrophage?
fibroblast. No, look, macrophage is here. And what is always characteristic for macrophages, these are these filopodia protrusions from the surface of the cell. Okay? So what type of cell is this one? This is mast cell. Do you remember mast cell? What is mast cell doing in the connective tissue? What are its, for
Is it
example,
inflammation during allergy response?
yes, exactly. So what about the this one, for example, plasma cell? This is plasma cell. And what you have to remember when you try to identify, oh, what I have here. So cells, Uh, in the case of plasma cells, that nucleus of plasma cells contains very well-developed heterochromatin and euchromatin. Yes, and they are arranged in clock face style or wheel-like manner. So, however, this is characteristic for plasma cells. What is this? In the center of nucleus of plasma cell, here, what is this? Or what is here too? For example, in macrophage, you can identify the same thing. What is this, guys? Nucleus. Nucleus is this one. Look.
Is it nucleolus?
Nucleolus. Nucleolus. So, why nucleolus is so dark stained? Do you remember? Have you studied this everything in? I think you have not studied in. You have not started cell biology, yes? Okay.
Not yet.
Yes. So look, please. Usually, nucleolus is not so dark stained because of prevalence of heterochromatin. No. So nucleus contains a lot of uh, RNA, and which is in very active state. I mean, the main function of nucleolus is ribosome production. So ribosome subunits. So here in this nucleolus, or here, for example, the intensive process of transcription is initiated, and a lot of subunits, I mean, protein subunits are produced in the nucleolus, subunits of ribosomes. Okay, so let's go to this image. Look. So what you see here, what is this cell? Look, this one. And that one. First, this one or that one. Look, okay, if I give you the,
They're fibroblasts.
yes, this is fibroblast. So fibroblast has this euchromatic nucleus you see here. Nucleolus is clearly visible, and cytoplasm of fibroblast is dark stained because rough endoplasmic reticulum is very well developed. The situation, the same is here, for example. This is also fibroblast. But what is here in this one? This is fibrocyte. Look, fibrocyte. They have much more thinner, flattened nucleus, smaller. They are much more smaller cells uh, in comparison with the fibroblast. And their nuclei are dark stained because the chromatin is tightly packed, and they are inactive, less active cells. Okay, so this, what type of tissue you see here? What type of tissue is on this slide? So this is loose connective tissue. And you see here collagen fibers. These are collagen fibers. And this not stained part is ground substance. So for midterm, you have to know content of the ground substance, how they are arranged in the ground substance. Please start to read the textbook because a lot of material you have to study, and step by step we can go through the most difficult and most complicated topics, re-explain it again. So please read the textbooks, okay? If you look at the presentations, and you have to do this, especially from presentations which will be uploaded on the drive as a set of micrographs for midterms. So I will do this today or tomorrow, and you have to work on these micrographs, okay? So this is connective tissue. Connective tissue proper. What are these cells? Look here, dark stained. Nuclei. This, for example, might be a lymphocyte. This too. Oh, sorry, here are two cells. Look, one and another one. And a lot of collagen fibers. What is here on this image? Look, this cell or that one or that one. Plasma cells. Plasma cells. So what type of cells mainly are described in the connective tissue proper are, of course, cells, blood cells, first of all, and there might be the different type of um, leukocytes might be there, macrophages, plasma cells, mast cells, and fibroblasts or fibrocytes. Okay, so here we have plasma cells. And look, plasma cells are also quite active cells. They have both, as here, heterochromatin, euchromatin, but very well developed rough endoplasmic reticulum. Tricia, what do you mean detailed and labeled micrographs? Look, this collage, this set of micrographs, I can include in your questionnaire, and my question will be, what type of cell is represented on micrograph 2? I think it is enough for this question. I mean, labeling and numbering. What do you mean when you write me, please, can we have detailed and labeled micrographs? So, okay, wait. Look, you mean organelles should be indicated there. Yes, guys, you know, during all these years, the histology was included in the life science one. And usually we start the histology with the cell biology, I mean with the cell. Now histology moves to the body system. It became part of body system, which is very logic and which is very integrative approach, but I forget that we have not studied the cell biology. That is why I ask the questions, and now I understand, Prisha, what you mean labeled. Okay. So look, guys, in all these four micrographs here, you see four different cells with a cell membrane, cell bodies. So you see here cell bodies. Look, for example, this cell, fibroblast. This is cell body, and it is surrounded, of course, with a cell membrane, and this thin line represents the cell membrane. You see nucleus in the cell. You see rough endoplasmic reticulum. Look, this is rough endoplasmic reticulum here, and nucleus, okay, with a nucleolus. The same is here in the plasma cell. We have nucleus, we have nucleolus, we have nuclear envelope. Look, this is nuclear envelope here, and it contains two leaflets usually. This is cell membrane here. Look, cell membrane. And this is cell cytoplasm filled with the rough endoplasmic reticulum, mitochondria. These are mitochondria, and ribosomes are also here. These dots, which are not clearly visible now, but if you use the high magnet, if you compare, for example, this image, I mean, the image of fibroblast with this one, look, this one, I will put it here too. Look, this is fibroblast in LM, light microscopy. This is fibroblast in TEM, transmission electron microscopy. Okay? And what you see here, you see nucleus. Look, this one is nucleus, and this one is nucleus too. In LM, you can see the euchromatin and heterochromatin too, of course. Look, these dark spots are in the nucleus is heterochromatin, and more dark stained part is heterochromatin, and more light stained is euchromatin. Nucleolus is in the middle, and nucleolus is here. So this is cytoplasm. Look, which is dark stained, but you can't identify here fine details. Even if the, for example, magnification will be much more higher than here it is, okay? So this is due to the resolution power of microscope. So content of cytoplasm is clearly visible in the TEM micrographs. For so okay, for that reason, in your micrographs, I mean, in your midterm questionnaire, only LM micrographs will be used. Okay, because we have not studied yet the cell biology, and we have not studied how to identify this different type of parts of cell structures. But I think this is the most interesting part. Okay, this you will have everything. But now at this moment, in the midterm, second midterm will be included only micrographs of LM, light microscopy. Okay, so loose areolar connective tissue with the fibroblasts and fibrocytes. Okay, again, this is connective tissue, and here proper connective tissue proper, which contains, beside the other cells, you can identify here very well the plasma cells. And what you have to know, what is the main function of plasma cells, and you have to know also how to identify them. Okay, first of all, please look at the nucleus and this arrangement of, look how the heterochromatin is arranged in the nucleus. Okay, so this is another type of micrograph, and here again we have the plasma cell with a nucleus, with a rough endoplasmic, with a cell cytoplasm, and you know this not stained part or less stained part is Golgi complex. Okay, so plasma cells, what are these cells? Mast cells, yes. And how we can identify them? Of course, using their morphological features, that they have granulation, which is clearly visible even in the light microscope. Okay, so what type of tissue is this? Reticular connective tissue. And here, these dark stained lines, black stained lines, these are the cells which, reticular fibers. Okay, please, can we have, oh, so we can different between different internal structures of each cells. Okay, so what type of connective tissue? This is reticular connective tissue. Okay, which is silver stained, and in the only in the silver stained tissue samples you can identify these fibers, reticular fibers. So this is lymph node, and here you see the reticular cells, which are actually the fibroblast like cells. They give rise to these reticular fibers, and this is adrenal cortex here, and usually these reticular fibers represent the collagen type 3. So when we are talking about the connective tissue proper, and even when we are talking about the extracellular matrix, so we have to know the components, main components of this matrix. So what they are, these main components. Do you remember main components of extracellular matrix? ground substance, fibers, yes, and also in extracellular matrix, you can identify always the fluid which is called interstitial fluid. So you have to know this, how this interstitial fluid is produced in the tissue and its main function, how the components of ground substance, I mean glycosaminoglycans, it's different type of glycosaminoglycans, and also types of protein fibers, of course, and main types, of course, and elastin fibers, collagen fibers, uh, and how they are produced there. Mm, have you studied in the life science protein synthesis translation, translation? Not yet. So you have not studied synthesis, how the synthesis of proteins takes place in the cell cytoplasm. Okay. What type of cells we have here? I think these micrographs have small resolution, and after zooming they do not look like very well, but okay, I will improve that. So this is fibroblast. This one is osteoclast. This one, plasma cells and mast cells. What type of tissues are here? Connective tissue, reticular connective tissue here, elastic connective tissue, and loose areolar connective tissue. Here, dark stained are elastin fibers. Pink stained are collagen fibers. So look here. On this image, we have collagen fibers with pink to red stained lines. You see them here. And these black stained lines are elastin fibers. Reticular fibers, yes, Prussia, I think all presentations I have uploaded on the drive, okay, on the Google Drive. All of you have access to this drive. I think if you have New Vision mails, you have access to the drives too. And here is embryonic connective tissue. Embryonic connective tissue. I could not find this presentation. Even now, please check now, Charlene. Maybe you will see it. So you see here embryonic connective tissue and which is mesenchymal tissue, and another one is mucous tissue. So we have here cells and cells here too. Look, mesenchymal cells which have tapered ends, and extracellular component contains a lot of collagen fibers and ground substance. The same is here in the mucous tissue. Mucous tissue is actually tissue which fills the umbilical cord. It is represented in the umbilical cord and also in the tooth pulp in the adults. So this is actually the mesenchymal like tissue too. Look, the cells, they are interconnected with each other and give rise to the jelly like structure. How? For example, this tissue is able to form this jelly like structure in the umbilical cord, which suspend the blood vessels in umbilical cord. Due to the glycosaminoglycans, which are highly hydrophilic, they attach a lot of fluid, water. That is why we have here this Wharton's jelly. So, and what is the main difference between the mucoid tissue and mesenchymal tissue? Do you know? We have mesenchymal cells in the mesenchyme tissue and in the mucoid tissue too. Both are mesenchymal cells. But in the mesenchyme, cells are able to differentiate further and give rise to the all types of cells of connective tissue. I mean, fibroblasts, fibrocytes, uh, adipose tissue cells, bone tissue cells, cartilage cells, and quite a lot of cells are included in these types of cells. So, but Wharton's jelly cells, they are already differentiated. So, and they can't differentiate further in umbilical cord. But you know, uh, during last 2 decades, these cells are used for transplantation. I have mentioned this, and or for different to substitute different injured cells in the human in the individuals, so in human body. That is why the umbilical cord now is these cells are kept and in appropriate conditions. But to differentiate in different lineages, I mean, differentiation of these mucous cells, the appropriate conditions should be, of course, created. I mean, in vitro. So, do you know what in vitro means? In vitro. In vitro means outside the body, in lab, in tube, in lab, in, yes, okay, so outside the body, okay, in artificial conditions. This might be the tube, this might be the petri dishes, etc., etc. Okay, is clear this difference between mesenchymal cells and mucoid tissue cells? Both they derived from mesoderm. Both of them are mesenchymal cells, actually, but the mesenchymal cells of mesenchyme are able to differentiate in different types and give rise to different types of connective tissue cells, but cells in the mucoid tissue, they are already differentiated. Okay, so another set, okay, look again, back to these images. What type of tissues we have here? All of them are tissues, connective tissue proper, and this is loose areolar connective tissue proper, but loose areolar, okay? Connective tissue proper, but reticular connective tissue with a prevalence of reticular fibers. Connective tissue proper, but with prevalence of elastin fibers, elastin connective tissue, okay? So, and look, another type of cells, another type of tissue, first of all, adipose connective tissue. And here the situation is much more easier because cells of connective tissue are easy identifiable. And when, for example, you have a set of micrographs together, and adipose tissue is really easy to identify. So, but what about this is white adipose tissue. I'm talking about the white adipose tissue. But what about the brown one? Look, brown adipose tissue. This is brown adipose tissue. And this is each of this figure, each of this profile, sorry, is profile of connective brown adipocyte. And what is characteristic for white adipocytes and brown adipocytes? So white adipocytes have clearly visible one big droplet of the lipids. This, I am talking about this white one. And the brown adipocytes have a lot of droplets there. You know, look, this is TEM image. TEM image which represents these cells in more details. I mean, brown adipocytes, for example. You see here, look, this is cell profile, whole cell. Look, whole one cell. And you see here several lipid droplets. Another one are not visible because in this plane of section are included only these droplets. In the center is nucleus. Look. And cytoplasm, this is cytoplasm. Everything what surrounds the nucleus is suspended in the cytoplasm. So in the cytoplasm, you can identify lipid droplets and you can identify mitochondria. Look, these small round shaped profiles are mitochondria. Plus, you can see here cross transverse section through the capillaries. Look, this is red blood cell, RBCs. One of these cells is surrounded with 1, 2, 3 capillaries. It means that this type of tissue is highly vascularized. All these features, I mean, lipid droplets, mitochondria, capillaries, or vascularization provide much more dark staining of this tissue. Look, capillaries in the light microscopy, how they look like. And these are longitudinal sections. Look, longitudinal sections. These are transverse section. Okay? Hopefully you understand the difference between this um, longitudinal and transverse sections of the cells. So another TEM image, and you see here 1, 2, 3, 4, 5, and 6 parts of brown adipose cells, and only one cell is fully represented on this image with a look, cell body, cytoplasm, nucleus first of all, surrounded with the cytoplasm, and the cytoplasm is filled with droplets, lipid droplets, and mitochondria. Okay, this is capillary again. This one and here that one. So each of these cell, I mean each of these brown adipocytes or that one looks like this cell or that those cells in the TEM images. Okay. Um, Look at this image. This is these are lipid droplets. Here we have parts of two adjacent adipocytes. Look, this is part of one cell, this one. And this one is part of another cell. And you see here the, wait, sorry. You see here the cytoplasm. Look, if you look at this image, you see that the cell is filled with the lipids and small rim of cytoplasm surrounds this cell. So this small rim, this dark line actually, is represented here with a much more higher magnification and with a much more higher resolution power. That is why you can see here mitochondria, look, and you see small lipid droplets, mitochondria again, okay, look, mitochondria. You see the cell membrane, and what you see else here are dots in the extracellular matrix. Look, this part is extracellular matrix, which contains dots of collagens, because if we see here dots, they are in transverse section. And if we see lines, even short lines, it means that these are longitudinally or obliquely sectioned collagen fibers, okay? So small part of white adipocytes. This is the cytoplasm, a rim of cytoplasm, this dark stained blue rim. which contains collagen, oh, sorry, which contains the mitochondria, a lot of quite high amount in this small part of cell, and here too. And beside these big lipid droplets, we see lipid droplets of different sizes. And these lipid droplets are in the fusion process with this big one. Look here, 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 they undergo fusion with each other and with this big droplet. Okay? So here is small part of fibroblast cell. Okay, is everything clear with this image and with this one? Look, guys, here on this, look, we see here or here um, much more, or here or there, much more amount of cytoplasm. This is cytoplasm of white adipocytes. And you know why we are able to see this much more amount of cytoplasm? Due to the plane of section. So the plane of section in this case goes through this surface, through this thin rim of cytoplasm. Beneath, of course, is other part of cell which is not visible, which is behind this cell, and which contains this big lipid droplet. Is it understandable what I say? So why you see here much more big amount of cytoplasm, this is due to the plane of section. Plane of section which goes through this surface, which is covered with the surface of the cell, which is covered with the cytoplasm. Okay? Is it understandable? Through these parts. So you know that the cell is three-dimensional structure. Oh, look. Do you remember the image of white adipose tissue, which I have shown you in the histology guide? Look this one. I'm talking about this one. Connective tissue. Look, so white adipose tissue in the light microscopy. Images which go of the transverse sections of the cells, and this is TEM image and only small part. This one is scanning electron microscopy. So when the plane of section goes very superficially and includes only the cytoplasm, in this case, you will see much more amount of cytoplasm like in this case here. Okay? If you go look, so plane of section through surface, then it goes deeper. So the small part should be included of the lipid droplets, yes, beside the cytoplasm, like this one. Look, like this one. When we go deeper and deeper, I mean sectioning of these cells, deeper, deeper, and deeper. In this case, of course, the profile of the cell will be such profile, which contains the adipose or fat lipid droplet and small part of nucleus which is located peripherally, and small amount of cytoplasm, small rims of cytoplasm. Okay, is it understandable, everything what I have told now, what I have tell you now? I mean, images of adipocytes, how they are represented in LM micrographs and TEM micrographs, and even in the scanning electron micrographs. Is it understandable?
Yes.
Okay, so another type of cells which we have in connective tissue specials. So another type of tissues, I mean, first of all, cartilage. So with the, I think with the brown adipose sites also is everything clear. I am talking now only about the morphology, about the structural features. I'm not talking about the function. Of course, you have to know functions, their functions very well. And why for, because actually their structure determine their function. So why the brown adipocytes have so many mitochondria? White adipocytes have also quite big amount of mitochondria, but if you compare the brown adipocytes with the white adipocytes, brown adipocytes have much more. So why? Because brown adipocytes are involved in thermogenesis, and thermogenesis takes place in the mitochondria. The heat is produced in the mitochondria, and white adipocytes also, they store, their main function is storage of fat. What for? What for they store the fat? Fat is the main source of energy. That is why they have quite a lot big lipid droplets, quite a lot of mitochondria too. So I think um, regarding the cartilage cells, look, or here, cartilage is specialized type of connective tissue. And what we have here, chondrocytes, yes, the cells, main principal cells of cartilage tissue are called chondrocytes. So what type of structure they have? So what are the main morphological features of the cartilage tissue as a whole tissue, and the main features of its cells? So the first of all, the cartilage has three subtypes. Yes. So in cartilage is type of tissue which provide the um, what for is cartilage? How do you think? For example, hyaline cartilage or fibrocartilage or elastin cartilage. They are quite strong type of tissues, and at the same time they are very flexible. So due to the content of extracellular matrix. And in the cartilage, in what type of collagen we have in the connective tissue proper? Collagen type 1. Collagen type 3 are reticular fibers. Collagen type 2 is represented in the cartilage. And in the cartilage tissue, in the hyaline cartilage, this is hyaline cartilage, we have cells which are grouped. They give rise to the isogenous groups of the cells. So we have here several different layers in this tissue sample. So what is the difference between these tissues? Look, this is dense connective tissue. This is growing cartilage. And between this dense connective tissue and growing cartilage is perichondrium. So perichondrium, which contains the mesenchymal cells, mesenchymal-like cells, which are differentiated into the cartilage cells or chondroblasts. Look, here are chondroblasts. And then later chondroblasts differentiate and give rise to the chondrocytes. So chondroblasts are able to divide. And cells in perichondrium, they are also have capacity to divide. That is why the perichondrium is the main source of cartilage growth. Usually this perichondrium is where? On the periphery of cartilages. Yes, cartilage is surrounded, covered with the perichondrium. And these perichondrial cells proliferate and differentiate into the chondroblast cells. Chondroblast cells are able to proliferate further and they give rise and differentiate into the chondrocytes. Even in the tissue, chondrocytes are deeper layers of the tissue. Chondrocytes are able to divide, and that is why they give rise to these groups of cells, okay? Isogenous groups. What is this, for example? This part of tissue, extracellular matrix. Extracellular matrix is also this part which surrounds immediately the look, this dark stained part of extracellular matrix. This is also part of extracellular matrix. They contain different amount of collagen fibers. That is why they are stained in different, and glycosaminoglycans. That is why they are stained differently. Immediately when these cells start to produce the extracellular matrix, they became chondroblast cells and then chondrocytes. So dense connective tissue, which contains these pink to red stained collagen fibers. Plus you see here profiles, look, this blue elongated profiles. They are fibroblasts, of course. Look, the collagen fibers are clearly visible. And this is dense connective tissue because they are arranged in one direction. Here is the perichondrium, mesenchyme-like tissue, which gives rise to the new layers of cartilage tissue. So this is a positional growth of cartilage. Growth on the periphery, via periphery. Okay? So new layers of collagen tissue are added to the peripheral part of collagen, of cartilage. In the middle, in the deeper layers of already differentiated cartilage, the cells are able to divide, but they are hampered in this extracellular matrix. That is why they can't, they are grouped, okay? And they also provide growth of connective tissue. But this growth takes place in deeper layers, almost in the middle part of cartilage. And this is interstitial growth. So a positional growth via the periphery and interstitial growth via the growing process from center to periphery. This is growing process from periphery to center. Okay? Is it understandable? Difference between the appositional growth and interstitial growth. Is it? Maybe you have questions. Please switch on your microphones or write me your questions.
Sorry, could you explain the difference between the interstitial growth and the oppositional growth again?
Yes, sure. So for that reason, I will show you another image. Look. Look, this is a longitudinal section through the hyaline cartilage. This is trachea, part of trachea, and you see that this cartilage is surrounded with a different type of tissues. If we magnify this image, look, magnify more and more. So look what you see here. You see cartilage. You see the connective tissues, dense connective tissue. You see also here the lining, epithelial lining of trachea. Here is, for example, also loose connective tissue. Look, adipose tissue with a loose connective, areolar connective tissue. How you can identify this loose areolar connective tissue? It usually contains the small amount of, look, usually contains adipose tissue, and the collagen fibers are not, they are arranged chaotically. They are in different directions. But this, look, I'm talking about this. This is loose areolar connective tissue, white adipose tissue, and usually loose areolar connective tissue is represented in the organs with together with the white adipose tissue. Here you can see the dense connective tissue. Look, can you see it? Look, this one. Dense connective tissue with collagen fibers arranged in one direction. And look, magnify this. Look, pink, red stained collagen fibers and fibroblasts or fibrocytes. white adipose tissue, collagen fibers, and this is areolar connective tissue. Okay, so dense connective tissue and perichondrium. Okay, look, perichondrium. This one, growing cartilage here and differentiated cartilage there. Okay, so perichondrium. Now I minimize the image again. Look. And what you see here, perichondrium, growing cartilage, and cartilage, differentiated cartilage itself. So this perichondrium here, perichondrium contains cells which are able to proliferate different chondroblast cells and give rise to the chondroblast cells. So these chondroblast cells here, initially they looks like the fibroblasts, you know. They are elongated, tapered, and then they step by step became differentiated into the chondrocytes. So when the chondroblast cells start to produce extracellular matrix, it became the chondroblast, and chondroblast then is differentiated into the chondrocyte. Look, you see here 3-cell group which contains 3 cells. Here is much more amount of cells. Here too. So these are dividing cells. They cells which have capacity to divide, but the extracellular matrix hampers them. They are not able to move and to allocate separately from each other. That is why they give rise to the groups of cells. This is growing cartilage layer, which contains chondroblasts. And new layers of cartilage are added in on the periphery of cartilage. I mean, here or there. This is appositional growth. But if you look in the deeper layers of cartilage, look, magnify it, you can see the group of cells too. Look, groups. And these groups of cells contains several cells. It means that they are also derived from one cell, which and they are able to divide. Okay, but again, they are not really able to separate from each other. That is why they are represented in a groups, but they are able to produce extracellular matrix. It means the growing process takes place in the deeper layers of the cartilage too. That is why this growing, this type of growth, I mean, on the periphery of cartilage, from periphery to the center, is appositional growth. And growth process, which takes place in the deeper layers of cartilage, in the middle on this micrograph, I mean, here, this is interstitial growth, okay? So the new cells, new layers of extracellular matrix, and it means new layers of cartilage tissues are added in the deeper layers of cartilage. This is appositional growth. This is, sorry, interstitial growth, and this is appositional growth, okay? Is it Now, understandable difference between the appositional growth and interstitial growth. Is it?
Yes.
Tarun, you write that it is not understandable. So, what especially is difficult to understand for you? What is your question? Can you formulate the question? The last one, what is the last one? So, growing process. Look, um, Prisha, maybe you can explain, Tarun, what is happened here on this image. What is the difference between the appositional growth and interstitial growth? Can you, Prisha? Where are you, Prisha? You are disappeared.
Yes, I'm here.
Okay, please, can you explain what is the difference? Can you say, Tarun, your colleague, what is the difference between the interstitial and appositional growth of cartilage?
The interstitial growth pattern is the one that happens inside the deeper layer of the cartilage, while the appositional one is the one that happens near the surface layer.
Surface layer. So look, Tarun, please. This is small magnification. And you see the cartilage in the middle. This dark stained one is cartilage. And you see the cartilage which is surrounded with a different type of tissues. And first of all, if you magnify, you see that cartilage is surrounded with a growing cartilage. So this is differentiated portion of the cartilage, and this is growing cartilage, not finally differentiated, okay? So this you can identify in all parts of cartilage. You look everywhere. So cartilage is surrounded with a perichondrium, okay? This is perichondrium here, okay? This growing layer of cartilage is included into the perichondrium. And perichondrium provide new cells, precursor cells for cartilage cells. So these precursor cells here, they looks like mesenchymal cells, or sometimes they looks like fibroblast cells. They differentiate and give rise to the chondroblasts. Chondroblasts. Look, you see here chondroblast cells. Look, which undergo differentiation, and step by step that they proliferate and give rise to the group of cells. So this, for example, this cell is able to proliferate, but it is not yet divided. This here, these cells is group of cells which is derived from the such type of dividing cells. And because they already start to produce the extracellular matrix, their daughter cells are hampered in the isogenous groups. So these cells give rise to the new cells, I mean this chondroblast, and new layers of extracellular matrix. It means that they give rise to the new layers of cartilage tissue. And this everything takes place on the periphery. So these new cells are derived from perichondrium. And new layers are added to the cartilage, but this is on the periphery. In the middle, in the deeper layers of cartilage, cells are able still to divide. That is why we have here groups 2. And they give rise to the new layers of extracellular matrix. So this growing process takes place in the deeper layers of cartilage. This is called interstitial growth. Okay? Is it understandable? No, Tarun. Is it?
Yes, ma'am. It is
Okay.
understandable.
In this case, so what type of tissue you see here? Look, this one. What type of tissue is this? White adipose tissue. This is, look here. This is loose areolar connective tissue. Look, the pink stained collagen fibers, which might be longitudinally sectioned or transversely sectioned. And you can see here also fibroblasts. Look, if we magnify these cells here, it looks like a fibrocyte. Look, dark stained nucleus and light stained cytoplasm. But this one here is definitely fibroblast. Okay, so loose areolar connective tissue. And here, loose areolar connective tissue and dense connective tissue. Dense connective tissue. Collagen fibers, fibroblasts. Look, this is definitely fibroblast. This one too, this one, for example. And perichondrium, look, in perichondrium, this fibroblast-like cells, they give rise to the new chondroblasts. Chondroblasts start to produce extracellular matrix. At the same time, it divides. They are able to divide and give rise to the groups. And new layer of cartilage tissue is formed. Okay. So the cartilage cells are allocated in the lacunae. So one cell per one lacuna. Okay. The lacuna is indentation in the extracellular matrix, place where the cells are attached to the extracellular matrix. The same we have in the bone tissue too. Do you remember bone tissue? Okay, this is, we'll go to the bone tissue. So you have to know the differences between different type of cartilage tissues. I mean, fibrocartilage, elastic cartilage. Look, this is elastic cartilage. And this is ear, external ear. And what we have in the extracellular matrix These are fibers, elastic fibers. Look, red stained elastic fibers. Okay, these are chondrocytes. Elastic tissue has the perichondrium too. But what is in the fibrocartilage? In the fibrocartilage, we have no, look, this is epithelium, this is skin. In the fibrocartilage, we have no perichondrium. In the fibrocartilage, you can see the collagen type 1 together with the collagen type 2. And you can see here also fibroblasts. Look. Look. So fibroblasts. These are chondrocytes and these are fibroblasts. Collagen fibers stained pink and elastin fibers. Okay. Look. Collagen fibers. Pink or red and blue stained are collagen type 2. Red stain is collagen type 1. So actually the fibrocartilage is mix of collagen cartilage, fibrocartilage and hyaline cartilage. Okay. This is fibroblast here and all other cells are cartilage cells. Cartilage cells, look, they all are cartilage cells. Look, fibroblast. Bone tissue. What type? You have to know the types of bone tissue, how they are um, actually developed. You have to know these processes: process of remodeling of bone, process of bone osteogenesis, intramembranous and endochondrial osteogenesis. Okay, so these are the two different types of processes which provide the formation of bones and also reshaping of the bones. Okay, and type of cells which are represented in the bone tissue. What type of cells we have there? So you see here this schematic image of bone. And what we can identify are, look, different type of cells. So of bone tissue. So all these cells, actually, these are different pathways of cell differentiation. Initially, we have mesenchymal stem cells. And these mesenchymal stem cells are able to differentiate and they give rise to the osteoprogenitor cells. So these osteoprogenitor cells are allocated on the surfaces, outer surfaces of the bone and inner surfaces of the bone. Okay? So on the outer surfaces of the bones, these osteoprogenitor cells differentiate and give rise to the osteoblasts. So the same is here in the inner surfaces. Osteoblasts are formed. And these osteoblasts, both of this outer and inner population of osteoblast cells, is able to differentiate and give rise to the osteocytes. So the osteoblast cells are able to produce extracellular matrix. Osteocytes are also able to produce extracellular matrix. They also are able to degrade this matrix in a small amount, which is very effective for calcium release when such type of need exists in the human body. So these osteoblast cells, beside the osteocytes, give rise to the periosteal cells or cells, bone lining cells, or cells which are kept as a precursor cells of osteocytes, but they are kept on the surface in the periosteum. Okay, these osteoblasts, which line the inner surfaces, they are beside the osteocytes, they are able to give rise to the endosteal cells. Again, precursor cells for osteocytes, but they are located on the inner surfaces of the bone. They line the trabeculi also. So in the spongy bone. So all surfaces in the spongy bone are lined with the endosteal cells. And periosteal cells are represented in the periosteum. Periosteum is layer of the tissue which has the same function as perichondrium has. Is cartilage vascularized or not? Cartilage.
No.
No, it has no, it is avascular. Bone is very well vascularized. Do you remember this Harvesian canals, canaliculi, lacunae, how they are interconnected? They actually perforate the mass of this mineralized bone tissue, and they give rise to the very well vascularized, very hard, firm type of tissues. So with the cells, mesenchymal stem cells, and with the different pathways of its differentiation, I think everything is clear. Is it? So what is the difference between the periosteal cells and endosteal cells? They are allocated on different surfaces of the bone. Yes, periosteum is on the surface, outer surface, endosteum is on the lines the inner surface of the bone. They are derived from mesenchymal cells, together with the osteoblasts, together with the osteocytes. Regarding the osteoclast, osteoclast is differentiated from blood cell, precursors of blood cells, granulocyte or monocyte progenitor cells. They give rise to the inactive osteoclasts, and inactive osteoclasts are inactive before they became activated by the osteoblast cells, you know. So very interesting relations are between the osteoblast, osteocytes, osteoclast cells. Actually, the bone growing process is process which includes process of bone osteogenesis plus remodeling of the bone tissue. So osteogenesis, we have two main types of osteogenesis. One is endochondrial and another one is intramembranous. So what has happened when we have intramembranous osteogenesis? We have a dense layer of mesenchyme. Mesenchyme undergoes condensation, and mesenchyme cells in the mesenchyme differentiate into the osteoblast cells. And osteoblast cells start to release the collagen fibers, and this densely packed mesenchyme looks like a real membrane, you know. And then they start to produce the osteoid. So what is osteoid? This is new, not finally differentiated layer of bone tissue. Okay, look, somewhere in the um, during the embryogenesis, look, we have here fetal phase. Look, formation process of the fetal phase. And you can see here the osteoblasts. Look, these are layers, look, spicules of bones. Look, this, irregularly shaped, okay? Or here, mandible and maxilla, future mandible and maxilla. So here in the middle, we magnify it and we will see the, look, mesenchymal tissue, a lot of mesenchymal tissues. Look, cartilage. And here cartilage is covered with the perichondrium, and it undergoes a positional growth and it undergoes interstitial growth, both of them, okay? But here, look, mesenchymal tissue, very well vascularized. And where we have the, okay, it should be here. I want to show the, look, bone tissue. So intramembranous ossification, membrane-like structure of mesenchyme, dense layers of mesenchyme, and they give rise to the osteoblast cells, this mesenchymal cells. Look, osteoblast cells, and osteoblast cells produce the osteoid from this side too. Look, osteoblasts, osteoid, osteoid, osteoid. And you know, when the osteoblast cells, and in the middle here, we all have already ossified tissue, bone tissue. Look, this one here with the osteocytes. These are already osteocytes. But again, this is fetus. Okay. Um, you can identify here also this different type of bone tissue which are or mature and immature. For example, woven tissue. So this one here, beneath the um, layers of osteoblasts is osteoid, which is actually the not the fully differentiated bone tissue. This is also not fully differentiated. The main difference between the look mesenchyme, okay. So cartilage. Mandible and maxilla. Look, both of them are clearly visible here. Bone spicules, cartilage, which later undergoes ossification and gives rise to the bone tissue. So when the mm, look here, we have cartilage, as I have already shown you, cartilage, and later this cartilage will be transformed into the bone. So bone, septal bone. Also, you can see here the ethmoid bone will be developed here. Look, and nasal conchae too here. So they all look, these cartilages, they are initially formed and they give rise to the initial model of the bone, and later this initial model of the bone undergoes of the cartilage. This cartilage undergoes ossification. Initially, bone model from cartilage is formed and then it undergoes ossification. So the ossification here is very interesting and complicated process. The cells of cartilage, look, I will show you this. They undergo hypertrophy and they die. After the dying, they give rise to the small spaces. This lacunae became empty. And then these lacunae fuse each other and give rise to the primary areolae. Look, I will show you this. How this process is conducted in the during the embryogenesis. Look, this is model of cartilage, model of bone, future bone. Initially, mesenchyme is condensed here and differentiate and give rise to the cartilage model. Then this cartilage contains the extracellular matrix, cartilage extracellular matrix with the chondrocytes. Look, chondrocytes, cartilage cells. So later, this cartilage matrix undergoes calcification and the cells die and give rise to the empty spaces. So these empty spaces fuse each other and give rise to this primary areoli. Look, they are called primary, the spaces are called primary areoli. They are separated from each other via the calcified matrix. Look, the bars of calcified matrix. On the another stage of chondrogenesis, the blood surrounding mesenchyme, and you have seen this, how intensively this cartilage is surrounded with a mesenchyme. So this mesenchyme forms here the blood capillaries, and blood capillaries, blood vessels invade the cartilage tissue. They, from one hand, they form the perichondrium and perichondrial it is perichondrium, but perichondrium is transformed into the later periosteum, and they form the periosteal layer, which provide blood capillaries for the bone tissue. So you see here invasion process of the blood by the blood capillaries, and together with the blood capillaries, blood vessels, the mesenchymal cells, new mesenchymal cell population invades this empty spaces and covers the surfaces of these bars. Look, of these mineralized, these blue stained are calcified bars. Look, calcified bars. So they became osteoprogenitor cells, and then the osteoprogenitor cells are transformed into differentiated into the osteoblast. Osteoblasts immediately start to produce new layers of bone tissue, osteoid. And step by step, this mineralized cartilage tissue is covered with the layers of bone tissue. Okay. Is it understandable?
Yes.
So in this case, is everything clear, guys? The difference between the and whole, as a result, this whole cartilage tissue became bone tissue. And the perichondrium is transformed into the periosteum, which has fibrous layer and which has cellular layer. 2 layers has the periosteum. Okay, now you have another, you have to take a break, even few minutes. Guys, if you have some questions, please write me. The syllabus of the midterm is uploaded on the drive. Everything is indicated there. I will upload the set of micrographs too. So feel free, write me if any questions will be arised. So
No,
see you next, meet you next week. I can't see you. but midterm syllabus is hard to find. Why it is hard to find? It is uploaded on the drive. You have to go to the folder medicine. I mean, I am talking about the drive, Google Drive. You go to the folder medicine shared with me on the left side. Do you remember this? Then medicine, and in the medicine first semester, and in the first semester body system 1, and in the body system 1 histology and embryology together. Okay, so in the search you can write, for example, medicine, or you can write directly first semester. It's up to you. Spring, you mean spring semester? It is indicated with this. I think no. It, you have to find out the folder medicine, then first semester, and then body system 1. In body system 1, histology and embryology, or it might be only histology written on the folder, or histology and embryology. It does not matter. And you can see there are 3 folders in this subfolder. So this 3 folder: PPTs, midterms and final exams, and textbooks. Only midterm 1? I will check. I will check because I have uploaded yesterday or 2 or day before yesterday. Okay, I will check, and if it is not there, I will upload. Okay, so any other questions? Okay, guys, you have anatomy now, or I know that another lecture you have now. Okay, meet you next week. Please write me questions. Bye, guys. Bye. Thank you for your attention.